next talk, and that is on clinical utility in polygenic risk scores. I'm Dr. Chantal Barbe-de-Villiers from Public Health Genetics Foundation, who's going to tell us about this.
Great. Good afternoon, and thank you to the organisers for giving me this opportunity to speak here. I've found the day really interesting, and I look forward to the discussions we're going to have in the breakout sessions. So I'm from the PHG Foundation. It's a policy and healthcare think tank, and I'm going to come in with a bit of a different perspective of looking at polygenic scores and their potential clinical utility. Oh, now my slides won't change. There we go. So the work I'm presenting today is from a report we at the PHG Foundation recently released on polygenic scores and clinical utility. For the purposes of today, I'm going to give a brief introduction to the concept of clinical utility, particular of healthcare tests, how evaluation frameworks and regulations consider clinical utility, and then finally the various perspectives on clinical utility of polygenic scores, including the proposed application of them in healthcare. So this work has evolved from previous work of ours, where we noticed there was considerable discussion and debate about the potential value of polygenic scores, where discussions have referred to numerous different proposed applications of polygenic scores, as well as definitions of personal and clinical utility. With all this debate, we saw a need to disentangle what was meant by utility of polygenic scores. We used a method of drawing from in-house expertise, literature review of both academia and grey literature, and semi-structured interviews with experts on polygenic scores. This report provides a background on key topic areas relevant to considerations of clinical utility, as well as a synthesis of the debate with respect to the utility of polygenic scores. So what are we looking for? What do we need to consider? When we talk about using polygenic scores in healthcare, we're talking about genotyping an individual, applying a polygenic score model to calculate that individual's polygenic score, and then interpreting the score. So for example, determining where they are along a distribution and what that means for them. To do this, we would need to have a polygenic score-based test. This test is what we're trying to understand the clinical utility of. So we need to understand how the clinical utility of tests in healthcare is determined. Understanding and using clinical utility to determine if a test is ready for implementation in healthcare is not new. So a review of how utility is viewed and assessed for tests was done as part of the report. So taking a step back, what is a test? A test is described as the use of an assay within a specific disease context, in a particular population and used for a particular purpose. Healthcare tests take a wide variety of forms and vary in their complexity, purpose, providers, and users. An assay is a method for determining the presence or quantity of a component, and the test is a procedure that makes use of an assay for a particular purpose in a particular context. The method used to analyze a substance in a sample is considered the assay, whereas a test is the use of an assay within a specific context. So as an example, and to provide context, the purpose is part of the test, where an assay may not change, but the intended purpose can change the nature of a test. For polygenic scores, apart from genotyping an individual and requiring an assay to get that information, we also have the role of the polygenic score model, that is part of the testing process. So meaning that the model can be considered the assay. The risk model is a mathematical representation to predict outcomes or a predictive algorithm. They can be either simple or complex. There is also potential of these models being transformed to tools, and tools can take a variety of forms and are mechanisms through which risk models are utilized to enable individual level testing and risk scoring. And they can include a mechanism to provide feedback to the user. The test is the procedure that makes use of a model and or its tool, so the assay, for a particular purpose in a particular context. Models and tools have the potential to be used in different contexts, so each could be considered the assay. It is important to understand these different concepts for the purposes of evaluation and regulation. Each has different requirements for each of these elements. So how is utility of tests considered? The concept and understanding of utility has no singular definition. The fact that they have no clear singular definition and that clinical utility can be considered in a narrow or broad sense means that definitions made are often contentious and context dependent. Conceptually, clinical utility refers to the usefulness, benefits, and harms of an intervention. However, opinions about utility are subjective. Clinical versus personal utility are broad terms that we constantly kept coming across. They're used to make an assessment as to the value of tests, and although discussion discerns a difference between the two concepts, they are interlinked. We ultimately use the term clinical utility to encompass both these concepts. The utility of healthcare tests has differing perspectives, and depending on the perspective, emphasis is placed on a particular outcome. A wide range of groups have an interest in utility. We just heard about insurance companies. These include the individuals undergoing a test, healthcare professionals, healthcare laboratories, advisors to healthcare systems where they're providing information on guidelines, or assessment units that evaluate evidence. There's also payers from individuals, hospitals, or departments of health, and of course regulatory bodies. Each of these groups may place greater emphasis on different elements of clinical utility. Healthcare professionals may assess utility to determine if the test will be of benefit in helping them make a decision about a patient in their care. Healthcare payers may place greater emphasis on determining value based on improvement in the current process, reduction in costs, and if it will answer a question at a cost they are willing and able to pay. Different decisions on utility can be reached on the same test and with the same evidence depending on who is making the value judgment. Such different groups may also adopt different mechanisms to assess the evidence with respect to utility, depending on their perspectives and needs. This is where evaluation comes in. So evaluation is a technical exercise used to enable healthcare decision makers to assess the evidence base for a particular test. A number of evaluation frameworks exist, including the frameworks for genetic-based tests. One that many of us might recognize is the ACE model, the process for evaluation of genetic tests. The intended audience of an evaluation differ, so frameworks more suited to each may exist. They may be for guiding policy decisions, for example, as a cancer screening framework, or to provide guidance for patients, as well as others impacted by the tests. The spectrum of utility and the elements considered within these frameworks varies, and few readily define the evidence required for clinical utility to be satisfied. Broadly speaking, though, genetic test evaluation frameworks aim to assess whether the test can accurately and reliably measure whether a variant is present, if it accurately measures or predicts the presence, absence, or risk of the clinical disorder being investigated, the positive and negative impacts of carrying out the test, cost of testing, and the usefulness of the information retrieved from the test. You also need to consider that evaluation of the assay or the test can take place. Evaluating an assay, in this case the model or tool, can be restricted to validating a particular methodology, so measuring a biomarker. This may demonstrate that a particular method used to analyze a substance is reliable and robust within a range of conditions. Evaluating a test is a more complex undertaking that encompasses wider considerations related to the use of the test and its value in addition to the technical performance. In addition to evaluation, it's important to understand how an assay or test might be regulated and how clinical utility is viewed in this setting. In order for an assay and a test to be used as intended, they must be shown to be safe and effective by complying with regulations. Occurs at different levels. For example, it could be the test, the laboratory, or the person offering the test. The environment is dynamic and currently in flux with Brexit and the introduction of new directives. For novel assays and tests, it may be challenging to identify the nature, quality, and quantity of evidence required to ensure sufficient safety and effectiveness to place a test on the market. The primary way in which a device might qualify as a medical device under the new directives is if it is intended for medical purposes. More tests used within health systems will qualify either as medical devices or in vitro diagnostic devices, depending on the manufacturer's intended purpose. The degree of scrutiny applied will depend on the nature of the test, how the test will be used, and the nature of the user. It's also important to realize that evidence of utility is not explicitly required for regulatory approval. So regulation is a legal requirement and evaluation is recommended technical exercise. There is, however, overlap between the evidence generated for regulatory processes and healthcare evaluation, which can lead to misunderstandings about their utility. They both are important components of evidence-based decision-making and provide a mechanism for systematic evidence appraisal. Similar kinds of evidence is needed between them and provides a structured way of thinking, allowing for more comprehensive considerations around utility. However, different decision makers may place differing emphasis on different aspects of utility. Different decisions may be reached with respect to utility and/or implementation of a test. And even when tests are considered to have utility, this may not lead to a decision to implement it within a healthcare setting. Decisions with respect to clinical implementation are likely to be influenced by a broad set of factors, including other competing priorities, level of impact, infrastructure requirements, opportunity costs, the availability of resources, clinical and personal outcomes, cost effectiveness, feasibility, and test delivery. The list goes on. Differing emphasis on these factors will depend on needs for the test, which can lead to contrasting decisions on utility, despite having access to the same evidence for a particular test. So a lack of clarity on the characteristics and purpose of polygenic score-based tests can be a significant limitation to determine the level of evidence required prior to their implementation. So how can utility be demonstrated? Here is a process to demonstrate utility with steps that can inform various aspects required in regulation as well as in evaluations. Currently, the majority of effort for polygenic scores has been on the analytical validity with demonstrations of assay performance, with speculation of their utility. What is also required is clinical validity and implications of the test use for management of disease. Utility is a multidimensional construct covering effectiveness and cost effectiveness, as well as the psychosocial, ethical, and legal implications of an intervention. So this was a quick basic introduction to clinical utility of healthcare tests. Next, we wanted to understand how polygenic scores and their utility was being viewed. This is a highly debated area that has much contention. We wanted to understand why and what the reasons for this are. Based on a review of the literature, as well as interviewing key stakeholders, we observed a few key issues. There are a number of variable factors that were influencing decisions on the utility of polygenic scores. The fact that decisions about utility are tied to value judgments and priorities means that it is an inherently subjective concept, and we were seeing this in the debate of polygenic scores, and is related to differing perspectives and individual viewpoints. The evidence base is currently mostly from academic research studies, rather than through comprehensive assessment or evaluation of specific applications that are considering specific care pathways. When both the specific context of a polygenic score use and the perspectives of those contributing to the debate were being taken into account, there is broad agreement over areas of potential use for polygenic scores. However, there were differing views held as to the readiness and suitability for implementation in healthcare. There were areas of agreement and consensus, for example, that genomic biomarkers, including polygenic scores, are not considered intrinsically different to any other type of biomarker. It is not possible to make any overarching claims about polygenic scores across the board and must be considered in relevant context of use and conditions specific, so the disease being investigated. Also agreed that within a healthcare setting, the use of a polygenic-based test must make a difference to clinical or public health decision-making to have utility. that some form of evidence is needed, although there is debate of which the level of evidence needs to be, and that randomized controlled trials might be the ideal way to generate this evidence, but are unlikely to be feasible in all circumstances, and there are limited alternative options, and that they are useful alongside other biomarkers as a part of a comprehensive risk assessment. So these were areas where there was agreement, but there were also areas where there was disagreement and challenges, and mainly varied based on perspective. There was the ability of genomics to contribute to prediction of complex traits, with concerns about hype and genomic exceptionalism, as well as the way in which information from polygenic scores was being interpreted. The relevance and the validity of polygenic score research, generally because it's currently mainly in academia at the moment, we also observed this to be an issue of the distinction between a model tool and test, and the need for development of polygenic score-based tests. The interpretation of the utility in relation to polygenic scores, where the utility is defined as the likelihood that the score will work as a test, and its associated intervention will lead to an improved outcome. There are challenges when considering the effectiveness of the proposed interventions as part of the utility of polygenic scores. For example, where the intervention is behavior change, there was deep debate for utility. The effectiveness of behavior change based on receiving genetic information are challenging. Considerations for these types of interventions varied significantly, depending on perspective and the understanding of the field, as well as the quality of the intervention. Effectiveness of interventions were also debated, as not all preventative strategies are benign, such as pharmacological interventions or surgical procedures, which are more controversial. Even seemingly simple decisions like increased screening may result in false positives with significant economic cost to society and unnecessary stress to the individuals. All these debates about interventions were being convoluted with the discussion of utility of polygenic scores. Finally, the evidence base required to demonstrate utility in different contexts, the nature and the thresholds of the evidence required to establish that a test fulfills a specific purpose was highly debated. Some believe any degree of improvement in discrimination is sufficient, whereas others wanted higher degrees of improvement before considering it to be useful. Standards are being introduced, but some agreement on which performance metrics need to be considered still need to be agreed. Different clinical decisions require different burdens of proof. Suitability will vary in each scenario, and the importance requires consideration of the context. Lastly, there are differences in expectations about the purpose of doing a polygenic score analysis, where expected outcomes may not just be clinical endpoints. It will be important to evaluate the outcomes that matter to decision makers. A risk-based intervention needs to be appropriate, accessible, workable, and acceptable. This is why clarification on intended purpose is required. So the importance of intended purpose clarification provides context and purpose of a test, which are key in evaluation and regulation, but also for the decision makers and individual user perspectives. Assessment of whether a test has value, so utility, is linked to the intended objectives of the test. The nature of the applicable regulations also depend on the characteristics of the test and the intended purpose and user group. Clarity on utility and discussion of polygenic-based tests can benefit from clarity on intended purpose. There are different uses that are being proposed. As we've seen today, there is a few of them as well. And a few of them that we came across were polygenic scores used as part of risk tools. This is seen as the most likely use of polygenic scores, where they may stand on their own as discrete predictors, or they could be combined with other risk factors in an existing or novel risk prediction model to provide a combined risk estimate. The main issue is around the extent of improvement in performance of a tool and how it is measured. Another potential application was polygenic scores as a screening stratification tool. This had very varied views, often relating to the utility of screening. Polygenic scores are being considered here where they can either be part of an existing screening program, a novel screening program could be introduced based on polygenic scores, or act as a mechanism to triage for or in screening programs. There was also polygenic scores for refining risk prediction in Mendelian disorders. This is thought to have clinical benefit, but there is concern, as we heard today as well, about the interplay between polygenic and rare variant risk. The last one is highly debated with early life screening for risk of common diseases, where there was often a lack of clarity and definition of what was meant by early life. Different potential scenarios where it was going to be used and proposed. Also, the different utility it would have if it was for an individual versus population health. So in conclusion, there is broad agreement that polygenic scores as a biomarker could have potential in informing different healthcare scenarios. The added value for polygenic scores is likely to vary for different use cases. Judgment on the clinical utility of polygenic score analysis requires a clear understanding of what information it provides and how this information is intended to be used. How polygenic score analysis as a novel test is approached is key to assessing utility in healthcare pathways, as well as the regulatory requirements governing its generation and use. What are the next steps that we could do? So far, significant effort has gone into evaluating the performance of polygenic score models. Attention should also turn to evaluating the tests as test systems that are based on these models. Mechanisms for evaluation and assessment of utility do exist and cover numerous aspects that are being hotly debated. Evidence now needs to be generated to demonstrate utility that is disease and context specific. Research into how they might be used in specific care pathways is also vital. Both require clear articulation of the proposed application, the specific clinical context, and the population in whom the test is to be offered. Without this information, we can't determine their clinical utility. Effective assessment of utility cannot focus on polygenic scores as an undifferentiated whole. To deliver the best outcome for both individuals and the health system, an assessment should be made on each potential test based on the use of a polygenic score model. So I just have to thank the whole team that was involved with writing this report and the work that went into it, particularly Salmi Morty, who led the project. And if you are interested, please do go and check the report on our website. And thank you very much. I do think there might be some time for questions as well.
Great, thank you very much, Chantal. So as Manir said, I've taken over the chair midway through your talk. So thank you. I thought it was a great overview of clinical utility, very thought-provoking, certainly for me, and the challenges we face, I think, in going from where we are now to bringing things into clinical care. There was, I think, one question in the chat so far. There's sort of mixed in amongst the insurance discussions. There was a question for Chantal from Sandy Starr. So unless I've missed it, the polygenic oligogenic distinction made earlier isn't part of the PhD report. Is this just a terminology issue or an area of confusion?
It's more terminology, probably. We would probably have pulled it under polygenic scores, and it's more the broader concept of clinical utility using pooled genetic variants to determine risk of an individual developing a disease. So yeah, it would fall under the report.
Yeah. Okay, so some questions are now pinging in from Alex Stoney. So how many PRS and also, sorry, so many PRS and also so many potential use cases. How does society ever do all the clinical trials required to demonstrate utility for each case? Good question.
Yeah, I think this is the excitement around polygenic scores. You know, genetics providing information for common diseases is something we've all hoped for. And now that it's showing potential, it's very much going to be figuring out what is the best way of assessing these and determining what's the best one to be using. And figuring out what do we need from a polygenic score in a specific context and situation to determine that it is useful. And it's true, there's lots of them out there. There's a lot of them out there.
So I mean, you alluded to levels of evidence earlier, and you talked about RCTs, but it's probably fair to say we can't undertake RCTs of every score in every use case. So did you come to any conclusion around levels of evidence and what we need to be able to implement these?
Guidance on what evidence would be suitable is good, but also I suppose key use case scenarios where the evidence has been proven, and then we could potentially extrapolate it to different situations. But we need at least some of that evidence to start being generated, and then we can start thinking about how it would fit within other scenarios.
Okay, question from Vincent. How do you quantify the fact that one genome-wide test provides info on many diseases? Doesn't that make the utility assessment challenging? How to use existing frameworks given the very specific feature of PRS?
These are all questions we need to figure out, I would say. And it is one of the big questions. And it's, you know, are we going to go in for a genetic test and have a PRS risk assessment for every disease done? Or is it going to be very disease case specific scenarios that we're going to land up having? Is how we're going to deal with this in the future. But I agree that, yeah, in theory, we could have a polygenic risk score done for every disease that we've got a decent GWAS on at the moment.
Yeah. Okay. So a couple more questions, and we'll make these the last two before lunch. So from Gary Ford, thanks for the overview on clinical utility. The National Screening Committee is highly conservative on genetic testing for cardiovascular disease. For example, they've not approved testing for familial hypercholesterolemia in children and parent cascade testing, despite the very strong evidence base about intervention from cholesterol lowering. They want CRCTs, which won't happen, so they're likely to be highly conservative about any PRS for screening. Comments?
Even in this type of scenario where you've got good evidence, it doesn't mean it's going to be implemented. So figuring out what is still needed, okay, they're clearly asking for RCTs, and then determining what alternative we could be using. But yeah, it's difficult, and figuring out when a test should be implemented and understanding the decision makers' choices on why they would implement one over the other. It's important to build strong cases to say this is going to work.
Yeah, okay. And just seeing the last one, there wasn't a question, it's more of a plug for a paper, and actually flagging that we've been having similar debates for a long time in pharmacogenomics world around, well, we can't test every drug gene pair in an RCT, so what level of evidence do we need? And I think by and large we didn't necessarily come to a definitive conclusion on that either. So we are entering into difficult territory, aren't we?